Hello students, welcome back to the biology class. So far we have covered the topic of sex determination, and we have understood now that there is something called as cytoplasmic inheritance. Cytoplasmic inheritance involves more contribution from the mother side or the maternal gamete, or you can say female gamete. That's why it is also called as maternal inheritance, because the cytoplasm, when we talk about the gametes, the female gamete contributes maximum, or in some cases the contributes the complete cytoplasm to the zygote in the future to develop. That's why the cytoplasm of the female gamete will become the zygote cytoplasm. That's why the characters which are controlled by the cytoplasmic genes, which in the class we learned before as the plasmon. So plasmon includes the chloroplast as well as the mitochondrial genetic material. So this material will pass on the characters to the next generation. That's why it is called as cytoplasmic inheritance. Remember, in this case the reciprocal cross result will be always different, because the result will be according to the cytoplasm of the female, not according to the cytoplasm of the male. Now we are going to start a very interesting topic, and that is how mutations arise. The topic starts like variations. So variation is, we all know variation is the key to adaptability, variation is the key to evolution. So NCERT also says that there are two methods of bringing variation. One is mutation and the other is recombination. In the last class I showed you the previous sheet of the notes, because in the beginning of the chapter we have learned somatogenic and blastogenic variation, and that time I explained to you discontinuous variations and continuous variation. See, whenever there is gametogenesis going on, we all know that crossing over will occur. So crossing over is a process which will cause recombination. So recombination is a continuous process of variation. But remember, in recombination, only shuffling or reshuffling of the genetic material takes place. Nothing new is added to the population genetics. But when it comes to the topic of mutation, mutation is a discontinuous source of variation, because it does not happen every time there is gametogenesis, but whenever it happens suddenly, and then it can be a good mutation or bad. But remember, maximum mutations are bad, and maximum mutations will be recessive in nature. So they will not show themselves that easily until unless they become homozygous condition, because they are recessive. So mutations are basically of two types. Mutations can arise in the genes, or mutation can arise in the chromosome. So please remember, when we say gene mutation means the structure of the gene. So we all know genes are segment of DNA, and segment of DNA means a sequence of nucleotides, or you can say polynucleotide. And when I say sequence of nucleotide, we all know in 11th class we have done that nucleotides are made up of phosphate group, ribose sugar, because we're talking about DNA, ribose sugar, and 4 type of nitrogen bases, A, T, G and C. Now the only variable component in the whole DNA is the AGTC, that means nitrogen bases. So the sequence of these bases are going to be fixed. If this sequence changes, even if a single nucleotide changes, that means the single nitrogen base changes, then it can cause huge problem in the genetic material, and you will call it as gene mutation. So gene mutations are the mutations at the level of the nucleotide. Please remember that. But when we talk about chromosomal mutations, chromosomal mutations are either the portion of the chromosome, that means some part of the chromosome would be missing, or something happens to the portion of the chromosome or the arrangement of the genes. And the second method possible for the mutation in the chromosome is the number of the chromosome could be changed. So accordingly they are called as chromosomal aberration for the structure of the chromosome to be mutated, and the number of the chromosome, it will be called as genomatic mutations. Please remember, Chromosomal mutations also include genes because genes are also a part of chromosome. But remember, if there is a genic mutation, if there is a chromosomal mutation of such type, it is possible genes may have remained normal. So that means genic mutations are a part of chromosomal mutation, but chromosomal mutations are not a part of genic mutation. I hope this point is clear. Now, the chromosomal mutations can be either the structure change or the number change. So let's start with the first one now. The aberrations in which there is deletion, duplication, inversion and translocation. There are 4 ways the structure of a chromosome can change. So the first one is deletion, and it can be of 2 types. Either the terminal portion of the chromosome is missing, or otherwise the in-between or intercalary portion of the chromosome is missing. Now we all draw chromosome in this manner. So if I put the sequence of the genes in this manner, so if the last portions are gone, it is called as terminal deletion. If anything in between is missing, it will be called as the intercalary meristem. So I have picked up the easiest numbers just to explain this concept. You can see in this diagram, I have drawn a hypothetical chromosome which carries A, B, C, D, E genes on the one arm. Okay, one more thing. This is a chromosome because we are talking about a interphase chromosome. Remember, if it enters into S phase, the amount of DNA will double, but number of chromosome will remain same. So this is the basic diagram of chromosome because we only want to show you the arrangement of the genes on it. So this is the longer arm, this is the shorter arm. P and Q are the names for it. So A, B, C, D, E genes are present on the larger arm or the longer arm. F, G and H are present on the shorter arm, and this is the primary constriction or the centromere.